Good morning, students. Today I'm going to talk about the last part of physiology of retina, that is the metabolic activity in retina. The metabolic functions of the retina. The metabolism correlates with its blood supply in the retina. Outer retina comprising the photoreceptor, the layers of rods and cones, and the retina pigment epithelium has a high metabolic activity and receives most of its blood supply from the choroid. While the other part, the rest 8 layer, the metabolism of the inner retina is supplied by the retinal circulation and is much less demanding of higher energy supply because the most of the activity is happening on the first 2 layers only. Basically, other than I say, only the layers of rods and cones. Other 8 layers are basically acting as a transporter because, as I said in last class, all are neurons only. So only they transmit the electrophysiological signals. Coming to the glucose metabolism, despite of having the highest rate of aerobic glucose consumption by any tissue, a large proportion of the glucose utilized is converted to lactate. This is a common metabolism activity we know already. The lactic acid production, oxygen utilization, and the glucose consumption are also highest in the presence of carbon dioxide bicarbonate buffering system, suggesting the role of carbonic anhydrase in the retina. Most of the glucose utilization in the retina is taken up by the photoreceptor, as I mentioned, because the highest energy is needed on that particular layer. So definitely the metabolism activity should be more over there. So that shows that it is more than 80%. The retina can also metabolize other substrates for ATP energy stores such as glutamate, glutamic acid, malate, and succinate. So apart from the glucose, they keep other substrate for it. Means if they need some energy, if the glucose is not there, so they can get it from either glutamate or glutamic acid, or malate or succinate. Retinal glucose is used to produce glutathione via the pentose phosphate pathway, which can be up-regulated under the condition of the oxidative stress. So if there is any problem in the oxygen, so they can take up that pentose phosphate pathway to produce the to metabolize the glucose and produce the glutathione. However, NADPH or glutathione store is also produced by the other non-pentose dependent system involving malate and isocitrate. Glucose, glutathione, and oxygen are all required for the generation of electrical activity, including the ATPase dependent dark current in the retina, as I mentioned, because once the light received by this photoreceptor layers around the cones, they converted that light into the electrochemical signals. So that is happening on the layers and rod and cones. So that's why they say that glucose, glutathione, and oxygen all required to generate the electrical activity in that particular layer. And definitely we need that ATPase dependent dark current. The retina is an insulin dependent tissue. One more important point here. So retina is an insulin dependent tissue. The glucose enters the retinal cells by the transport mechanism that are regulated directly by the extracellular concentration of the glucose rather than indirectly by the insulin. So that is most important part. So they regulate the concentration of the glucose in the cells. Glucose transports occur by facilitated diffusion via GULT1 and GULT3 transporter protein, similar to the glucose transport into the brain. The similar kind we have in the eye also. The few proteins are present in the brain to transport the glucose in the brain. So similar kind of here we have GULT1 gene. GLUT1 and GLUT3 transport components are present over there. Just remember for the short question, this will be very important. So GLUT1 and 3 are present in the cells of the blood retinal barrier where much of the transport occurs. These two proteins are present by the blood retinal barrier. That's the most important part because they only allow the glucose to enter to the retinal cells. So they only carry the things and bring to the cells. So that's why they're present over there. So most of the glucose handling described above, it's dealt with the retinal neurons. Obviously, there is a signal has to come, the instruction has to come from brain. So everything is dealt by the neurons. However, the molar cells are also likely to be involved in glucose metabolism in the retina. So other parts we get to see the molar cells are present in the retina, which also take a part on the metabolism activity. At least at the energy stores, since they contain high level of glycogen, especially in the species, in some species, not all, and that lack a retinal blood supply. In addition, lactate released by the molar cells can be metabolized by the photoreceptors, even though which is the end product of the metabolic activity, the lactate, which also metabolized by the photoreceptor cells. The photoreceptor in turn releases the glutamate, which is taken up by the and metabolized by the molar cells. Again, photoreceptors, which they release due to the metabolic activity, that is glutamate, also taken up and metabolized by the molar cells. Coming to the protein metabolism, so till that glucose or carbohydrate you metabolism, now protein metabolism. Many of the neurotransmitters required for normal retinal cells function occur as a free amino acid in the retina. So they need a free amino acid in the retina for normal activity for the neurotransmitter in the retina. So most of them are generated during the glucose metabolism in the citric acid cycle. The photoreceptor cells are the most abundant amino acid in the retina. Glutamate is a neurotoxic and is therefore converted by the retina to glutamine, and by the glutamine transferase localized to the molar cells. So glutamate, which is a neurotoxic, which is taken up by the, we have seen just now, molar cells, to convert it in the, and what is this? Taken up and converted and metabolized by molar cells. What they produce then? We can see glutamate is a neurotoxic thing which can damage the cells. So they are therefore the converted by the retina to glutamine by the glutamine transfer enzyme localized in the molar cells. The protein synthesis, as studied by such methods as the leucine incorporation, is the most active in the photoreceptor during the such process in the photoreceptor window. That is another important thing because otherwise it won't happen the photosynthesis. In addition to the main retina-specific proteins are present in the tissue. Yeah, definitely not only that only, we have so many proteins are present because initially what I did, the highest number of protein present in the retina than other than any organ in the body. So, for example, lamina is present in the basal structure, fibronectin in the interphotoreceptor space, matrix proteoglycan are widely distributed throughout the retina. Transin C is present in the extracellular matrix and is thought to play the role in preventing myelination of the retinal neurons. So each of them has some activities over there. Several growth factors are also present in the retina, such as insulin-like growth factor 1, that is IGF-1, acidic and basic fibrous growth factor FGF, that is called. Retina contains a high content of lipid also over there. So the predominant of the lipid in the retina are phospholipid, around 80% present phospholipid. There are also high content of the polyunsaturated fatty acid in the retina, especially in the outer segment, mainly the RPE or RAS or rods and cones. Lipid metabolism is varied and complex in the retina. Thus, in addition to the synthetic activity or the microsome exchange basis between the different lipid species occurs, while frequent acylation or deacylation reaction also occurs over there. Lipids are continually undergoing the degenerations via the phospholipases and the modification, including the decarboxylation and methylation by the appropriate enzyme. Coming to the last part, the blood flow to the retina. Yesterday we seen the what are the types of blood supply to the retina. So now it's auto-regulated. Means the retinal blood flow vessel caliber varies with the cardiac output in the to ensure that the blood flow kept constant. Alternatively, it has been interpreted as a meaning that the blood flow is varied according to the nutritional demand to the tissue. Retinal blood flow contains about the 5% of the total blood flow to the eye and majority passing through the choroid. Thank you. Read well. If you have any question, please ask me.